Hummingbirds of Mexico. For their health and ours, native to the Americas, hummingbirds aren't just beautiful to watch. They are environmental powerhouses. They are essential pollinators for countless plants and act as natural pest control by eating insects and spiders. But what happens when we change their world? My name is Lizeth Raygoza, and in this video I invite you to explore how human activity can change something invisible yet crucial. The bacteria living inside two species of hummingbirds that share our city. Our journey took us to western Mexico, to two very different places, the Guadalajara metropolitan area and the protected tropical forest of the Sierra de Quila. Both are home to hummingbirds, including species that live here year-round and others that migrate through. Some, like the rufous hummingbird, are even listed as threatened. These environments are increasingly shaped by human activity. Cattle ranching, large poultry farms, and intensive agriculture produce waste that can pollute rivers with chemicals and animal feces. In cities and rural homes alike, people often place artificial nectar feeders to attract hummingbirds. Feeders are not the problem themselves. In fact, they can help hummingbirds when natural flowers are scarce. But when they are not cleaned properly, the sugar ferments, fungi grow, and other animals, like bats, may visit them and introduce pathogens. In both the city and the forest, hummingbirds may be exposed to bacteria from contaminated water or poorly maintained feeders. Because they move constantly between flowers, feeders, and water sources, they can act as natural indicators of what is present in the environment. This led us to a big question. Could these birds be carrying bacteria that matter for public health? To find out, we focused on two local species, the broad-billed hummingbird and the violet-crowned hummingbird. We tested their droppings for 4 bacteria: Klebsiella pneumonia, an antibiotic-resistant bacterium that can cause pneumonia in people; Salmonella enterica, a germ that can jump from animals to humans, causing severe fever; Listeria monocytogenes, a foodborne bacterium especially dangerous for pregnant women; and Campylobacter jejuni, a common cause of food poisoning in humans. In the lab, we used a precise molecular tool called real-time PCR, like a genetic detective kit. We searched for the specific gene that tells us the bacterium is harmful. What did we find? We found that 63 out of every 100 birds tested had at least one of these bacteria. The most common was Klebsiella pneumonia, found in nearly half of all birds. Salmonella and Listeria appeared in lower numbers, and we found no Campylobacter. Species also mattered. The violet-crowned hummingbird was more likely to carry the disease-causing bacteria we studied, and it was the only species in which Listeria was found. The most important result was that risk depended on local conditions rather than broad habitat categories. We saw clear differences from one sampling site to another. Being close to a specific farm, the cleanliness of a feeder or the quality of the water in one part of a river could influence a hummingbird's exposure to bacteria, even within the same general area. This tells us something important. Hummingbirds are not the source of the problem. They are bioindicators, living signals of the environmental conditions created by human activities. When we see bacteria in hummingbirds, we are really seeing the consequences of polluted water, poor waste management, or dirty feeders. Their health reflects the health of the ecosystems we share. Simple actions can make a big difference. Cleaning feeders regularly, planting native flowering plants, and protecting natural habitats help reduce pollution from farms and cities. By improving the environments around us, we protect hummingbirds and ourselves. This research was published in the Journal of Wildlife Diseases. A huge thank you to all our collaborators, supporters, and co-authors for their incredible work on this project. Thank you for watching.